Hi, I will talk about this paper by Dr. Min Chen and et al. And this paper is again about HEMERA and zeroing in about how they degrade certain phytochrome interacting factors and how they transcribe their target genes. And honestly, this paper was really long. I think they could have divided the paper into two different papers. And actually, the first half is kind of about a complete digression from their title. But in order to just keep with the spirit of how they put it, I'll talk about everything here as quickly as I can. So in the start of this paper, they have certain mutants so they have mutants that don't have the interacting factor or HEMERA. And as you can see, whenever you don't have the interacting factor, HEMERA just doesn't, or the plant just doesn't respond to light. So they reason that interacting factor is epistatic to HEMERA when it comes to responding to light. However, HEMERA is important for this other function of keeping the plant green, meaning that they're important for, activating the chloroplasts in response to light. So some of the HEMERA goes to the nucleus to transcribe photomorphogenesis, and some of them go to the chloroplast to kind of start them up on their journey as well. Also, you can see in this diagram that the chloroplast genes are not activated when you take away HEMERA, and they are activated when you put in HEMERA. So now this is kind of the digression I was talking about, where they kind of dissect more biochemically how does the interacting factor bind to HEMERA. Here, with this immunoblot, they confirm that these factors do bind to HEMERA, and some of them bind stronger than others, like this PIF1 binds stronger than PIF3. They make these truncations on PIF1 to see which regions are really important, and they isolate two regions, this APB site, which you can see really binds really strongly, and then there's this BHLH site, which binds a little bit weaker to HEMERA, but these two sites seem to be responsible for their binding. You can see, though, that they really work best in tandem. If you take away one terminus of the phytochrome interacting factor, it just doesn't bind as well to HEMERA when you have both of them around. Even in this kind of weaker interaction, they're still stronger when you have both ends of the protein working together. So that's interesting to note. The C terminus is a BHLH site, which they say is a dimerization domain. They wanted to test if another kind of dimerization domain will do the trick. And indeed, we see that in this different kind of dimerization domain, the binding to HEMERA is still really strong, whereas in the monomer domain, it doesn't work. So really, it's just this dimerization factor that's important for the strong binding. It's interesting to note that the APB site on the phytochrome interacting factor also binds to phytochrome. So this region binds to HEMERA and phytochrome. They want to see if the exact same residues are also important. Here they crossed out, they had a mutant where they took away that residue that's important to phytochrome binding. But you see in this this GSTSA that particular mutation does not affect binding to HEMERA. So it's interesting to note that even though these two molecules need the same region, they don't need the exact same amino acids to bind to the interacting factor. So that entire section was kind of like the first half and digression of the paper. Now we begin the dissection of what they really talk about in their title, which is what sections of HEMERA are needed for degrading the PIF factors. They did this really complicated microarray, and they basically isolated two classes of genes that they think are important to study. The first class were upregulated in the absence of HEMERA, and the second class were downregulated in the absence of HEMERA. And they chose to zero in on this class B genes to study what regions of HEMERA are important for kind of the transcriptional activity of genes and for the degradation of phytochrome interacting factors. Inside this diagram, we have yeast growing on media, and there's a certain media stripped of certain nutrients. So they basically, the test is to see if HEMERA can transcribe certain genes in the yeast. If it does, the yeast will make those nutrients and keep itself alive. But if it doesn't, then we know that that section of HEMERA is not needed for transcriptional activity. So you can see in this section here that the N-terminus of HEMERA is not necessary for not necessary for transcriptional activity. So it's really the C-terminus of HEMERA that has transcriptional activity and that will keep this yeast alive. Again, they truncate the C-terminus into different sections. First of all, they isolate 6 possible candidate regions for the transcriptional domain, and then they truncate it to see which of them are really necessary for transcriptional activity. And it's this highlighted band here, the amino acids from 512 to 521, these are like the key region to do the transcriptional activity. You can see in the diagram on the right that without that region, the yeast just doesn't live. This particular region is they classify it as an acidic transcriptional domain, and the abbreviation they gave here is 9AA TAD, basically 9 amino acid transcriptional activity domain. Again, they see that these 9 amino acids are conserved in a whole bunch of species, from moss to higher plants, and really in particular is the 1st, 5th, and 9th amino acid that are crucial for the transcriptional activity. You can see on this graph on the right, if you take away any one of those amino acids or switch them to something else, the transcriptional activity just dramatically drops. We also note that there's this 4th amino acid that doesn't have as much dramatic of a change, but they chose this amino acid to mutate for all of their future kind of experiments on the protein. So here's some, So here's some class B genes and their expression levels. And you can see in the mutant of HEMERA, where that one particular amino acid was taken away, the expression level drops. Furthermore, in the same mutant, you can see that the interacting factors are not degraded. So the reason that that section of the protein is needed to transcribe those genes and also degrade the interacting factors. Here's some supplemental figures basically these are just proving that even in the mutant, you have plenty of HEMERA. You can see here that they have just as much HEMERA in the mutant, and they bind just as well to the interacting factor, but you don't have these phenotypes where it's degrading the factor and it's transcribing the genes. So really it's just one region that's essential for those activities. It doesn't have anything to do with the rest of HEMERA. Just that one region in HEMERA is essential for both of these functions. So they wanted to see now if a different kind of transcriptional domain will do the same job. And here they used one from a virus called VP16. And you can see in this graph that it doesn't do the same job. It just doesn't respond to light as well as the wild type. It's basically still as if you didn't have the protein. So it's really interesting to note that a different kind of transcriptional domain won't do the same job as the original kind.